Good morning, good afternoon, good evening, wherever you find yourself in the world today. Thank you for joining us. Welcome to the Vive Sense webinar entitled Human Augmented AI is Vital for Clinical Trial Endpoints. My name is Patrick Hankey. I am Strategic Business Developer at Vive Sense, and I will be your moderator today. I'm actually going to start by introducing our discussants. We have, not on video, Kate Lydon, who is our lead scientist. Dr. Lydon is an expert in monitoring physical activity and how that relates to chronic disease. And joining us on camera, we have Dudley Tabakin, who is one of the founders of the now 10-year-old Vive Sense. And Dudley has served as its Chief Executive Officer since its founding in 2010. I'm going to outline today's agenda. We'll roll into Dudley's presentation, and then we will open it up for a number of poll questions after the presentation, and we encourage people to send us questions if you'd like in the Q&A or chat boxes, and we will endeavor to answer your questions at the end of the meeting. So without further ado, I will hand over to Dudley. Thank you.
Thanks, Patrick. So as Patrick said, and thanks for the introduction, Patrick, and good morning, good afternoon, good evening to everyone. Thanks for joining us today. I wanted to just start off by thanking everyone for attending, and I want to say that I hope everyone is staying healthy in this time and staying safe and healthy, and we look forward to seeing all of you or meeting all of you soon if we haven't met you already, and definitely speaking to you further. So today I'm going to talk a little bit about human augmented AI from Vive Sense, and this is actually a presentation that was done in early March at the Tricon Digital Medicine Summit in San Francisco. That was at the start of COVID-19 outbreak, and San Francisco was being hit hard, and actually there weren't many attendees at this conference. So we felt that was really important information, and we should get it out there, and this is why we wanted to do this webinar. And also we think it's a good forum at this time to present information to all of you. All right, so first a brief introduction to Vive Sense. Vive Sense are a wearable sensor data core lab. We develop wearable sensor biomarkers, we bring them through clinical trials, and we do that in numerous ways. We have to be involved in all parts of the clinical trial and all parts of the study, from clinical study design, from how you're going to use a digital biomarker in the study, to sensor data, what's the best sensor to use, how do you collect the data from those sensors, how do you integrate that into your clinical trials. We are very interested in data quality control, ensuring that good quality data is obtained, good quality results are reported, and we're also capable of customized data analysis, because not every trial is the same. You've seen one trial, and you need to be ready for that. So Vive Sense clinical trials approach. The Clinical Trials Transformation Initiative, CTTI, put together principles of how to run clinical trials using digital health or digital medicine, digital technology. You can see on the chart on the left the details of some of the elements that are important: selection, data collection, FDA submission, inspection, data management, protocol design, and execution. And these are all really important. We know that the future of clinical trials is going to go towards digital technology. We see that even more now with COVID-19 outbreak, where you cannot necessarily bring your patients or your subjects into the clinic. A lot of more data collection will need to be done at home. In real world evidence will need to be used. But the CTTI and Vive Sense, we all understand that wearable sensor data, this data it's complex data, and we need to approach it in the right way. So the CTTI has this approach. And from that, we've developed Vive Sense's overarching principles to clinical trials. And those are those 4 principles that you see down the right side. Hypothesis driven is number 1. You know, we need to know what this trial is about, who's this for, what is the disease state, what are we looking for? Principle number 2 is that one size does not fit all, right? If we know that we can measure something in healthy adults, that doesn't necessarily translate to healthy infants or to disease state or specific disease state. So we need to be able to customize quickly. Good data is more important than big data at this time. Now, of course, there is a place for big data, and with this particular COVID-19 outbreak, you know, epidemiological studies, big data can be very important. But good data and understanding what is good data is very important as well, especially in a clinical trial setting. And then data quality and fidelity, everything has to obviously be managed appropriately. All the data needs to be appropriately audited and available whenever it's needed, and also under regulatory compliance. Okay. So in this presentation, I want to focus on that good data being more important than big data at this stage. And this is something that Vive Sense really concentrate on to ensure that quality results are reported and that digital biomarkers can be defined and validated. So I've got a little example here of something that we have done in Vive Sense. What you'll see on the left-hand chart is a period of data from multiple devices. We have an ECG trace, tidal volume, activity or motion being recorded. And then from that, we've derived the trend of the heart rate, which you can see is the blue line, third graph from the bottom. Sorry, second graph from the bottom. And then the bottom graph was the trend respiration rate. And we'll see on that chart on the left that it appears that there's an event occurring, right? There's an increase in heart rate, trend heart rate, there's an increase in trend respiration rate. And if we were looking at this data and trying to identify specific events, we might believe that this is an event. Something has occurred here that is relevant and we should be looking at it. However, when we look at the activity, we see activity as well occurring. And when we look at the tidal volume, we see a strange curve appearing there. And I'm going to use my mouse so I point at this. We can see this occurring here and this occurring here. And we might believe, and in this case, our Vive Sense AI believes that this is now artifact. This is not data that we can interpret correctly. And if we interpret it, we're going to identify an event that isn't an event. If we exclude this data, which we've done here, we can see that immediately our trend heart rate and our trend respiration rate no longer have this effect. There's no longer an event occurring here, according to, if we were looking for an event that occurred with heart rate or respiration rate. And so by cleaning out or identifying this artifact, and then removing it, we're actually improving the quality of the data. We're ensuring that good data is being analyzed. And this is, of course, is also done with a full audit trail and with regulated validation. All right, so we call our approach to good data human augmented AI, and that was the title of this webinar, so we'll get to that right here. So many algorithms out there, or AI, they are tuned. You have to have a trade-off between sensitivity and specificity. If you're just going to use AI, you're going to measure something like number of steps taken and or steps, and you might identify all the steps, and you'll trade off between sensitivity and specificity, and you may have an accuracy of 80%. Steps maybe wasn't a good example, but say you're measuring respiration rate, you may have a trade-off of 80% accuracy when you go that route. What we do in Vive Sense is we tune this for sensitivity. So we'll pick up a whole lot more false positives, as you can see this over here. We'll pick up more false positives over there, all right, but we increase the sensitivity, the specificity. Now we can use our expert analyst to adjudicate for the false positives and drive this back so that we get close to a perfect accuracy on these results. This improves our quality of data and ensures ensures that we can validate these biomarkers. All right. So in rare disease, we have done this a lot. We've done a number of trials in rare disease. This is one of them, spinal muscular atrophy type 1. This disease is a rare neuromuscular disease, causes loss of motor neurons and muscle wasting, causes diaphragmatic muscle weakness, which also causes obstructive breathing. There's 8,000 to 10,000 cases in the world now. You probably know there's at least 3 companies that are developing drugs for infants with SMA type 1. And we are involved in many of these studies. SMA phase 2 studies are being run right now. What we do is we measure chest and abdominal respiration from respiration bands or respiratory and plethysmography bands. We do that in order to identify the asynchrony between the chest and the abdomen. So respiratory endpoints of obstructive breathing are the phase relation between the chest and the abdominal breathing. If the chest is moving in one direction and the abdomen is moving in the other, then you have obstructive breathing. And this is cause for concern in infant SMA. You can also look at things like the percent of ribcage contribution to respiration. As infants grow or people grow, the contribution from the ribcage increases, and it's very low in the early stages in infants, and much lower even much lower infants with SMA. And we can also look at things like respiration rate and other respiratory measures. So in this particular study, and referring back again to good data, the good data part of it in the Vive Sense human augmented AI part, we use selection criteria and Vive Sense labeling and Vive Sense AI to identify valid breaths for analysis. We do this so we can reduce the false positives in the labeled data. We looked at an internal study looking at 61 subjects, 358 recordings, and we identified valid breaths for analysis. And I'll get into how we did that, what it means to have a valid breath in a moment. And we call these the labeled results. And we compared these labeled results to the unlabeled summarized results. So if we took the entire recording and we just averaged the data throughout the recording, we didn't clean it, we didn't clean the data, we didn't look for good data, we just used the entire data set. And so we had a comparison between that entire data set and our labeled, Vive Sense labeled data set. So first of all, valid breath identification. Why do we need to do that in SMA? Well, first of all, the SMA infants we're working with, the first problem is spirometry cannot be used. There are not other methods to measure respiration for a start. You know, these are infants, you can't have them do a forced respiratory maneuver for one. Secondly, you need to use a wearable sensor such as the respiratory bands, because you're looking for asynchrony between the chest and the abdomen. So you actually have to measure the changes in chest and abdominal breathing rather than an overall spirometry measure. Another reason we want to identify valid breaths is because infants cry and fuss. They move around, particularly those with SMA. They're having a breathing, obstructive breathing, and they're crying and they're fussing and they're moving about. And because of that, you have wearable sensor noise that is extremely high, and we want to identify only the breaths that are valid and good for analysis. Otherwise, we could just be analyzing noise. So the Vive Sense human augmented AI approach, we clean the data, we label the valid breaths for analysis, and then we follow standard operating procedures for our expert analysts to determine what is a false positive and what is valuable data. Having a look at the chart on the right, you can see this is actually an infant or maybe even an adult with normal breathing, and you will see the RC and AB bands, and the one in green, RC in red, AB in green, and you can see they're actually in perfect synchronicity, well, almost perfect, and this would be normal breathing. This would not be obstructive breathing. All right, so I mentioned that we identified valid breaths. How we do that? Well, we follow published work as a start, and we're looking for things like a closed conomed loop, which you can see in these charts over here. These charts over here, this is a conomed loop. It compares the rib cage to the abdominal breathing. And if it's a closed loop, then that's what we need. We don't want it to be open. No crossover the loop, so we don't want them to cross. And we also want a stable VT baseline with discrete peaks. And the VT baseline is this blue chart over here, and you can see stable baseline and discrete peaks is what you want. So now in this chart, we can see two breaths identified next to each other. And here's one that's been validated by one of our analysts, and here's one that they've decided is not valid. All right, and looking at the breath, you can see it's not quite the same as the other breaths. So from their perspective, they've decided that that was a false positive and have eliminated from this analysis. If we look at it closely, we can note that immediately, because of the length of this breath compared to the other breaths, that this would affect the respiration rate. If this breath was included, that would not be the normal respiration rate of this individual for this trial. So that's what we're applying the Vive Sense human augmented AI on. We're reviewing the valid breaths, the ones that may be false positives, and we're following those standard operating procedures. All right, so let's show you the results of that internal study where we looked at, as I said, the valid breaths that we had identified versus the overall average of the data. And what we'll see, first of all, in phase angle, we can look at the mean of the phase angle, and we can see 118.2 to 116.2, pretty much the same. Respiration rate, we can see there's a bit of a difference made. All right, when you look at all the data, you get a respiration rate of 54 breaths per minute. Remember, these are infants, they breathe at this rate. When we label the data, or we look for valid breaths, we get a rate of a little higher, 62. But the important part to look at is really the change in standard deviation. When we look at all the data, standard deviation for the phase angle, 32 degrees. And when we look at label data or valid data, we see a average standard deviation of 16.3 degrees, which obviously is half of what we're seeing. Looking at the charts below, the green line represents all the data, and all the dots below are the 361 trials, 358, sorry, sessions in which we've labeled data, and we can see that for the most part, they all fall below the green line, which means the standard deviation is less than what it would be for all if we looked at the same data in a single file for each of those trials. And you can see that happens in respiration rate and in the phase angle. The blue dots represent recordings in which we couldn't validate too many breaths. Less than 20 breaths could be validated. And so you can see some of those went above the line, but when reducing that number down to too few breaths, then it becomes difficult to perform better than average. However, but improving the accuracy in this way, as you can see in that previous example, we're improving accuracy, or at least standard deviation, by 50%. By doing that, we can reduce the required digital endpoint effect. So if we're looking at phase angle and we want a reduction in phase angle of 60 degrees, having a standard deviation of 16 degrees is much better, of course, than 30 degrees. If we can reduce that digital endpoint effect, we can reduce the subject sample size potentially. If we do that, we can save money on clinical trials. We can also speed up clinical trials. By doing this also, we're able to provide evidence for validation for the FDA and move things forward. So we believe in better science and improved outcomes. Here's a little graphic of the magic that happens, right? You put data in, and you've probably all seen something similar to this. Put data in, and then they have the black box where magic happens, and then suddenly there's a digital endpoint. I've crossed out magic happens because, as I've hopefully shown you now, magic doesn't happen. It's really about validating and doing good science in order to get the results that you need and the valid digital endpoints that can be used in clinical trials. I'll just take you through one more study that we're also working on and have worked on. That's in Rett syndrome. Rett syndrome is also a neuromuscular disease that affects girls mostly. It causes respiratory problems such as apneas and hyperventilations and hypopneas during wake. Similar approaches used here. We're identifying the events. In this case, this is Cheyne-Stokes breathing, Cheyne-Stokes respiration, which is very common in these girls. You can see the AI has identified Cheyne-Stokes breathing there and apneas along the way, and an analyst now can come in here and quickly review this data and decide whether that is valid or not. In this case, obviously it looks valid. By the way, the technology used here was similarly respiratory plethysmography, but this applies across the board to all types of sensors, whether it be gait sensors, wrist-worn actigraphy, ECG patches. The approach to finding quality data, to ensuring good data, is the most important for validating digital biomarkers in clinical trials. And that's the end of my presentation. And so thank you.